Eliminate infection. So infection will get removed by destroying the pathogen. Okay. So over here, you must remember that complement system takes part in both types of immune response, that is innate as well as acquired. So this is very, very important thing. It is not a part of innate, neither a complete part of acquired. It is a part of both. So you should not categorize it into innate or adaptive. We take it separately because it is a part of both, right? So it takes part, complement system takes part in both innate immunity and adaptive immunity. Okay, in both it is taking part, right? So we cannot separate it and put it in innate or adaptive. We should keep it separate from innate and adaptive always. Okay, so what happens over here is the complement system, it can get activated in two pathways.
Sir, sir, what is in the sixth line first word, sir?
Sixth line. Now I have to calculate the lines. 1, 2, 3, 4, 5, and 6. Destroy the pathogen. The complement system can be activated in two main pathways. In two main ways. Okay, so one way, if you see, one way is classical pathway and the other being alternate pathway. In the two ways, if you see, one is classical pathway okay, and the other being alternate pathway. Classical pathway and okay, in both it is taking part, right? So we cannot separate it and put it in innate or adaptive. We should keep it separate from innate and adaptive always. Okay, so what happens over here is the complement system, it can get activated in two pathways.
Sir, sir, what is in the sixth line first word, sir?
Sixth line. Now I have to calculate the lines. 1, 2, 3, 4, 5, and 6. Destroy the pathogen. The complement system can be activated in two main pathways. In two main ways. Okay, so one way, if you see, one way is classical pathway and the other being alternate pathway. In the two ways, if you see, one is classical pathway okay, and the other being alternate pathway. Classical pathway and alternate pathway. When we take the case of classical pathway, okay, and here you must remember that it is first and most potent way. First and most potent way of complement system. Okay, so over here what happens? IgG, immunoglobulin G, or IgM. So here antibodies, these are antibodies. IgG or IgM binds to an antigen. Binds to an antigen. Okay. at the surface of the cell, right? Antigen on surface of cell. See, antigen will be present on the surface. Okay. Next, this exposes the FC region. It exposes the FC, fragment of crystallization. In antibody, it will be there. We will see this when we talk about antibody, right? FC region of antibody. Okay, such that C1 protein means complement 1. Okay, C1 protein binds. Okay, there are 30 proteins we said, right? So C1 protein binds to the antibody when the antibody binds to an antigen. This is how it gets activated. So over here, antibodies are involved. Antibodies are a part of innate immunity or acquired immunity. We said B lymphocytes are involved in adaptive immunity, in humoral immunity, antibody mediated immunity. It is a part of adaptive immunity. Classification. Earlier we gave a classification chart, right?
Yes, sir.
Okay. This is the second pathway. Second pathway is the alternate pathway. Okay. So it is a part. It involves okay, certain natural immune response. Natural immune response. So production of antibodies will not be involved. So it is related to innate. Okay, so alternate pathway with respect to innate. Classical pathway with respect to adaptive. Okay, so here remember that neither T cells nor antibodies are involved. Neither T cells nor antibodies are involved. Okay, so in this case, what will happen then? Certain polysaccharides found on bacteria, okay, activate the system. See, students should have learned about the cell wall, cell membrane. Okay, cell wall and cell membrane. On the cell membrane, you remember there will be various types of polysaccharides, glycoproteins and all will be present. Okay, so like that, certain polysaccharides that are present on the cell wall, cell surface of the bacteria, they will activate. Okay, and this may occur instantly. Okay, this reaction, okay, reaction may occur immediately. Also, this also you can just keep in mind. Okay, the proteins over here, okay, the proteins may also create pores. Okay, like we discussed in the natural killer cells, right? So over here also, they may create a certain pores, okay, in the plasma membrane. As a result, the water enters, okay, and they burst, okay. And there is one more thing called opsonization. Opsonization, okay. Opsonization is the process of, it is the phenomenon of making a pathogen more susceptible to immune system. Means we are making it more available for the immune system to show its response. For example, let us say there is a pathogen, okay, on which our immune system is not showing so much of response, okay, or it is not showing very active response. In that case, certain proteins, what they do is, so over here, the proteins, they coat. Okay, so some components are
Phenomena of what is the another word?
the phenomenon of making. Okay,
Okay, sir.
making. Okay, so over here, what happens? Some components, okay, that is proteins only, na, in the complement system. Okay, proteins of complement system. Of complement system. What they do? They coat the surface of pathogen. Okay. So because of this, what happens? This will be easily phagocytized. Okay. It helps in easy phagocytosis. So easily it could be identified and phagocytosis will be done. Okay. So we can say that the pathogens are becoming more vulnerable, right? Because of the opsonization process. I hope you got this point now. Okay. See, complementing is nothing but we are like encouraging, okay, or making it feel good, right? So complement system, it is helping the immune system, right? Okay, it is in one way helping our immunological defense only, right? Okay, it consists of certain proteins. How many proteins? Around 30 proteins. Present where? Present in the serum, that is the blood. Okay, in the blood only you will have serum, right? What is serum? Can anyone define it? Earlier in the tissues topic.
Plasma without clotting factors. Plasma without clotting factors.
Without clotting proteins. Good.
Yes, sir.
So everyone remembers it.
Susceptible.
Huh?
So what do you mean by susceptible?
Susceptible means we are making it more prone. Okay, for example, susceptible in the sense vulnerable. We can in one way take vulnerable also. I even gave you an example also, right? If a pathogen cannot be recognized easily by the immune system, this complement proteins, they will go and bind, because of which it could be easily identified. Okay, so the pathogen becomes more exposed because of this, right? So it is becoming more susceptible so that it will undergo some damage, right? Now, the second part of immunity is acquired immunity or adaptive immunity.
Sir, so what is the use of serum, sir? It doesn't have any clotting factors, no cells also, no, sir?
See, it's not about use of serum. Okay, blood has water and all everything, right?
Yes, sir.
Okay, everything is mixed. Everything is mixed. So colloidal substance is formed. That is blood. If you remove one component from that, okay, you get different things. For example, if you remove all the cells, the remaining thing is plasma. Okay, in a plasma, if you remove the clotting proteins also, okay, then water along with certain albumin, okay, immunoglobulin and all is nothing but the serum. Whatever left over is serum. Okay. So whenever you need to transfer certain things which will not have the blood cells, okay, serum will be transferred. For example, antibodies. Okay, antibodies could be stored in serum. Okay, this is another advantage. Serum is nothing but the water in the blood only, na? Water along with certain colloidal proteins like albumin, immunoglobulin and all. Next one is adaptive immunity. Adaptive immunity or acquired immunity. Okay. Why we call it as an acquired immunity? Because we acquire it during the lifetime. Okay. So over here, it is gained after birth. Gained after birth due to exposure to pathogens, isn't it? And will it be specific or non-specific? It is a specific type. specific type. Okay, it is specific type. Okay, and over here, if you see specific type in the sense specific response, or we can say a special type of response will be there for a single type of pathogen. Okay, so every pathogen has different type of response. Every pathogen will have different type of response. Okay, so for bacteria, one type of bacteria, it will show one type of response. For another type of bacteria, another type of response. It is all on the basis of the type of pathogen. Okay, the type of protein or type of antigen present in the pathogen. Based on that, it will depend. Okay, and if you see over here, adaptive immunity or acquired immunity, it acts as the third line of defense, isn't it? First and second line of defense, innate immunity. The third line of defense, okay, it is the acquired immunity. Okay, so over here, it will recognize in this type, it recognizes okay, then if you see after recognizing, what it will do, it will attack, it attacks and kills the pathogen. Okay, it kills the pathogen, right? And this killing, it involves specialized cells, okay, or antibodies. Cells will be involved in cell-mediated immunity. Antibodies will be involved in antibody-mediated immunity. Okay, so over here, if you see certain properties of acquired immunity, okay, so properties, if you see.
Sir, what is the last line?
It involves specialized cells or antibodies. It recognizes, it attacks and kills the pathogen. Properties of acquired immunity, if you see, okay, it shows specificity. First thing we already said, right? Specificity. Okay, so individual response. Okay, so it will distinguish between the pathogens. It can distinguish between pathogens and show specific response. Okay, for different types of pathogen, different types of response will be shown. Okay, then diversity. Diversity is also seen, right? Diversity in the sense it can identify, okay, it can identify many types of pathogens. Many types of pathogens, isn't it? It can identify bacteria, if it is a virus, if it is a fungi, okay, or protozoan, okay, or a element. Different types could be recognized. And within the same also, like for example, in bacteria, different types of bacteria will be recognized in a different way. Okay, then there will be discrimination. Discrimination. Okay, means it can distinguish between self and non-self cell. Okay, self in the sense cells of own body. Okay, non-self cells means foreign cells, means any bacteria or pathogen that comes from outside. Okay, and when it distinguishes, what it will do? Because of this discrimination feature, it attacks non-self cell and protects self cell. Self cell is protected, non-self cell is attacked. Got it? Then there is a very important property, memory. Remember we said in the case of non-specific type, that is innate type of immunity, memory is not there. Okay, we just said, right? It has no memory of the previous attack. Memory, it is one of the important feature. Okay, so over here, a memory of the antigen is stored from the previous attack, from the first attack or previous attack. Okay, because of this, what happens? Because of this, what happens? When the second time, okay, it helps in easy and fast response. Easy and fast response during the second encounter. So whenever the same pathogen, second encounter of same pathogen, yes.
Sir, what is antigen?
You'll come to know about antigen. Don't worry. Okay, in further discussions, we'll talk about the antigens also. Okay, a complete antigen, an incomplete antigen, the antibodies. Okay, about all those things we'll be discussing. Do not worry. Okay. So over here, remember till here, right? It can show response, very easy response and very fast response and higher response during the second time. Okay, so based upon the response, you have two different types of responses. Primary response and secondary response. Primary response and secondary response. See, primary means whenever first time, okay, whenever the first time a pathogen enters, okay, it shows a primary response. It will initiate the primary response, okay, and this primary response is of low intensity. Okay, what I will do is a type of graphical representation I will give you first. So this graph, it is with respect to exposure, okay, and blood antibody level here. Blood antibody level, okay, and here time. Okay, so what happens is whenever there is a first encounter, okay, so when there is a first encounter, okay, first encounter, at that particular time, it will take so much time to show the response, okay, and the response will be comparatively less. Okay, such type of response will be shown. Okay, this is the primary response. Okay, because see, first time when the pathogen enters your body, okay, your immune system must identify, recognize, then produce antibodies and all, right? Okay, now when the same pathogen enters for the second time, okay, when the same pathogen enters for the second time, already a memory is created, right? Because of the first attack, okay, because of which what happens within a very short time, okay, within a very short time, the secondary response that is shown is comparatively very high. Okay, so antibody levels will be comparatively very high. And this is in general the case for IgG type of antibodies. Okay, during the secondary response, okay, more IgG type of antibodies. Okay, there is one more antibody over here. It is noteworthy. In this case, if you see, in this case, the IgM, if you see, it will be low only during the primary response and during the secondary response also. Okay, comparatively low. Okay, so this is for IgM antibodies. When we talk about the antibodies, now I will give you the number. By default, in our body, see, in our body, the highest amount of antibodies that are present are of IgG type. So during the secondary response, this IgG type of antibody can be produced in huge amounts or not? Definitely, because they can be high in number. IgM are not high in number. So this is the primary response. When the first time the body is encountering a particular pathogen, and this is the secondary response. Okay, when the second time the body is encountering a same pathogen. Okay, so here what I will do is I'll make a tabular form for it, for the response. Response, we'll take it in a, we said primary and secondary, right? Primary response and secondary response. So primary response, if you see, okay, it is an initial, occurs due to initial encounter. So when there is an initial encounter, okay, and secondary response, it occurs during successive encounter. Okay, so over here, primary response, is it of high intensity or low intensity? Tell me.
Low.
Low.
It is of low intensity, whereas secondary response,
High.
High, sir.
high intensity. Okay, the secondary response is of high intensity. Okay, and over here, the primary response, the time required will be relatively longer
More.
time. It takes
More, sir.
relatively longer time. Okay, and over here, it takes relatively less time.
So encounter means attack, no sir?
Yes. Relatively less time. Okay. Why it is taking relatively less time?
Sir, already the memory is filled.
Yes. Okay. So over here, what happens is in this case, okay, primary encounter, okay, the first encounter, it produces, okay, after exposure, okay, okay, immune system produces IgM. Okay, IgM, and this IgM will further decrease, and there is a gradual increase in antibody IgM and IgG also. Okay, and because of the production of this one, okay, there is a formation of memory cells. Okay, there is a formation of memory cells. The specific B and T cells. Specific B and T cells form memory cells. Okay, they will contain memory of antigen. It will contain the memory of the antigen, right? In the memory B and memory T cells, right? And they are stored in lymphoid organs.
Sir,
Sir,
is there any full form for IgM and IgG?
Ig is for immunoglobulin. M is the word used for it. Ig for immunoglobulin.
Yes, sir.
In blood, already we discussed this. Again and again, everything from starting we can't discuss, right?
Sir, when blood was being discussed, I was not in your batch, sir. I was actually in batch 2.
In any batch, it would be discussed. Immunoglobulin protein is present in the plasma, so it will be discussed everywhere. You don't remember, don't blame it on others.
Yes, sir. Sir taught in batch 2, sir.
Sir, I know about IG, sir. I want to know about M and G.
These are the suffix used over there.
Okay. Okay, sir.
So over here, the memory cells, presence of memory cells cause more production of antibodies in less time. Because here it need not go through all the process once again. Okay, and over here the secondary response is anamnestic response. Is anamnestic, means heightened response. Okay, anamnestic response. Okay, so a very high response will be shown, right? So these memory cells, it could last for days to years. Some memory cells could be there for lifetime also. Okay, some memory cells, they could be for lifetime also, right? Now, this immunity, adaptive immunity, we know that it is of two types. That is the humoral immunity and humoral immunity or antibody mediated immunity and cell mediated immunity, right? So about that we'll be discussing in our tomorrow's class. Okay.
But if someone upload this class also,
so God only can help them. With means they are amateur people, right? Not editing also, isn't it? Not editing properly. They should